Hey, what's up, everybody? We are beginning the respiratory system today. It's our first lecture on the respiratory system. So we got some big functions of it, including gas exchange, voice production, regulating plasma pH, odor detection, and then prevention of infection. So these are all major functions of the respiratory system. And we've got some major respiratory system events we're going to go over as well, including pulmonary ventilation, alveolar gas exchange, gas transport, systemic gas exchange, and then ever so briefly, cellular respiration. So let's start talking about pulmonary ventilation. Pulmonary ventilation is basically just a fancier way of saying breathing. And if we are breathing, we are exchanging air between our lungs. Can't spell breathing. That's an N right there. Exchanging air between our lungs and the atmosphere. Now we know in order for air to move, there has to be a pressure gradient. And when we are breathing in, air is moving because lung pressure is less than atmospheric pressure. And when we are breathing out, air is moving out because lung pressure is greater than atmospheric pressure. Now, obviously, the thing we're going to change when breathing in and breathing out is lung pressure. So when we breathe in, we need lung pressure to decrease, become lower than atmospheric. When we breathe out, we need lung pressure to increase, become greater than atmospheric. So that is pulmonary ventilation. We'll come back to this later on. We're going to come back to all these events that we're introducing. Then we got alveolar gas exchange. And that is the exchange of gases, particularly CO2, carbon dioxide, and oxygen, between lung air and plasma. So in order for oxygen and CO2 to move, there has to be a pressure gradient for those gases. So we know oxygen needs to go from the lung air into the blood. So oxygen's got to go from the lung air into the blood. So your pressure of oxygen in your lung air is greater than your pressure of oxygen in lung blood. And that's what's going to drive that movement of oxygen. Meanwhile, we know CO2 has got to go from the blood into the lung air. And so there's got to be a pressure gradient for that as well. The pressure gradient of CO2 has got to be greater in the blood, so greater in the blood, and lower in the air of the lung. And this is what's going to let us do alveolar gas exchange with these two individual different pressure gradients here. Okay. The third big event is gas transport. We got to transport CO2, we got to transport oxygen from our tissues to our lungs and vice versa. Now, most of our oxygen, 98.5%, is transported by binding to the iron, which is in the heme, which is in the hemoglobin, which of course is in red blood cells. So that's how we transport the majority, the vast majority, almost 100% of our oxygen. Now CO2, the majority of CO2 is transported in plasma as dissolved bicarbonate. So here is CO2 being made in a tissue cell. CO2 is diffusing into the plasma, diffusing into a red blood cell. Inside the red blood cell, CO2 reacts with water to make bicarbonate. The bicarbonate then leaves the red blood cell, ends up in the plasma. So most of our CO2, 70%, is part of this bicarbonate in the plasma. All right, we had alveolar gas exchange earlier. Now we got systemic gas exchange. This is gas exchange, again, particularly CO2 and oxygen, between the plasma and the tissue fluid. So I know CO2 needs to go from my tissue cells, from my tissue fluid into my blood. So my pressure of CO2 is higher in my interstitial fluid, lower in my blood. Meanwhile, I know oxygen's got to go from blood to tissues, so oxygen has a higher pressure in the blood, lower pressure in the interstitial fluid. All right, the final big event is cellular respiration, which we are not going to revisit. Unfortunately, that would be fun to do. Cellular respiration refers to how we break down nutrients like fatty acids, amino acids, sugars like glucose, in order to produce ATP, which is the energy currency for our cell. Cellular respiration primarily takes place in mitochondria. We are going to use up oxygen and make CO2 in the process. That's the only thing we're not going to revisit. We're going to revisit all the other big functions. But before we get there, before we do that, let's walk through the basic pathway of airflow. So I'm going to list everything, go through a couple differences, and we're going to individually go through all of these spots. So air is going to flow into your nostril, better known as your nares, to your nasal cavity, through your posterior nasal aperture, into your nasopharynx, oropharynx, laryngopharynx, through your larynx, trachea, main bronchus, lobar bronchus, segmental bronchus, then to smaller and smaller bronchi, then to a variety of bronchioles, then to a terminal bronchiole, then a respiratory bronchiole, an alveolar duct, and finally an alveolar sac. That is my basic inward path of airflow, and of course air can go in, but then air can also go out. Notice I got a couple different colors and other things going on here. The things in blue here, the things in blue are going to be your upper respiratory tract. So in blue, we got the upper respiratory tract. Whereas the things in purple, therefore, are going to be your lower respiratory tract. So upper respiratory tract nearest to larynx, lower respiratory tract, trachea onward to the alveolar sacs in the lungs. Notice also that I made some things bold and other things not bold. So the bold structures here, and I'm going to redo that and make it even more better. There we go. The bold structures here are what's called my exchange zone, also called my respiratory zone. So my exchange zone or my respiratory zone are those bold structures. And of course, with a name like exchange zone, that is where we are doing gas exchange. The unbold things, everything else, that is going to be my conducting zone. That is where I am moving air, but I'm not doing exchange. So 1 through 14, nares to terminal bronchiole, that is going to be my conducting zone, where I move air, but I don't do gas exchange. Notice terminal bronchiole is the last thing at the end of the conducting zone. That's why it gets the word terminal. Then my respiratory bronchiole, my alveolar duct, and alveolar sac, those are my three components of my exchange zone or my respiratory zone where I do gas exchange. So if I look in this picture, I can see the division between my upper and lower respiratory tract. Upper, boom, 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 down to the larynx, then below the larynx, trachea, and beyond, lower. I can also see the delineation between the conducting and respiratory zones here, okay? Conducting zone up to a terminal bronchiole, respiratory zone below a terminal bronchiole, where I'm doing exchange. And notice where I'm doing exchange, I have all these little alveoli here. That's where exchange occurs. So those are my functional divisions. Upper versus lower is a structural division. Conducting versus respiratory is a functional division. All right, let's talk about that conducting zone. Primary function is, of course, air transport. That's why we're using the word conducting. Secondary function, removal of the 3 Ps. What are my 3 Ps? Pollen, pathogens, and particles. Adding water, adding heat to air as well. That's my conducting zone. Now, most of my conducting zone is lined by what's called respiratory mucosa, which is going to be a pseudostratified ciliated columnar epithelium with goblet cells. And I see a goblet cell right here. Goblet cells, of course, make mucus. There are also going to be mucus glands in the underlying connective tissue. So I have some underlying connective tissue here. I don't see any mucus glands in this particular view, but there would be mucus glands in here with ducts reaching upward. Goblet cell and mucus glands make mucus. We see all the mucus right there. And of course, the cilia are up here, and the purpose of the cilia is to sweep the mucus, which will have traps things in it, to sweep it upward. Okay, that's my respiratory mucosa. It is in my conducting zone. Now, my respiratory zone, that's where I find alveoli. So every one of these little round guys right here, that's an alveolus. That's an alveolus right there. Got a whole cluster of alveoli as well. So alveoli are my simple squamous structures for gas exchange. I only find alveoli in my respiratory zone. I mean, that's what defines the respiratory zone. Respiratory zone contains alveoli. And I'm going to find it in three structures. Respiratory bronchioles. So this is a respiratory bronchiole. This is where we generally have lonely alveoli. Another respiratory bronchiole right here. And then I've got a tube made of alveoli. A tube made of alveoli is an alveolar duct. And then finally down here, I got a big cluster of alveoli. That is an alveolar sac. And that trio of structures, all of which contain alveoli, are going to make up my respiratory zone. Okay, I will see a transition in my epithelium. So I start with that pseudostratified ciliated columnar epithelium with the goblet cells. And basically got that up through my big bronchi. Then as I get into smaller bronchi and bronchioles, I go on to not a pseudostratified, but a simple columnar epithelium. And then as I get into smaller and smaller bronchioles, I get shorter cells, so it becomes more cuboidal. I start losing the goblet cells. I'm losing the cilia. Then eventually, by the time I get to the alveoli, I'm left with a simple squamous epithelium. So I'm going from a protective structure in my conducting zone, ultimately to an exchange structure in my respiratory zone. Makes sense. All right, let's now walk our way through the anatomy, starting with the nose. And in the nose, we have the external nose. We see the cartilage in there. The nares is the opening, the nostril. The singular is naris. Nares is the plural. Then there's a little space right here. This is called the vestibule. We have hairs in there. Nose hairs are properly called vibrissae. This is where you can have a booger in there. Vibrissae, nose hairs. And then if we keep going in, we got the internal nasal cavity. And this is where we're going to do a lot of the important nose functions. So first of all, though, we got to realize our nasal cavity is separated into left and right halves, separated by a nasal septum. The anterior part of the nasal septum is made of hyaline cartilage. Nice to have cartilage in the front because cartilage is more flexible and squishable than bone. Behind the cartilage, we have the two bony parts of the nasal septum. The purple one, number 2 right here, is the perpendicular plate of the ethmoid bone. And then this green guy here is the vomer. So those two make up my bony nasal septum, which is in the posterior. If we take the septum away, we can see the bony architecture of the nasal cavity. So here's the roof of the nasal cavity up here. I've got my frontal bone here. I've got my ethmoid bone here. And I got my sphenoid bone back here. On the lateral wall of the nasal cavity, I've got my maxillary bone right here. I've got my superior and middle nasal conchae, which are part of my ethmoid bone right here. Got my inferior nasal conchae, which is they are bones of their own. Got my palatine bone in the back right there as well. Now the palatine bone also helps me form the floor of my nasal cavity. That's the horizontal plate of the palatine bone. And then I got the palatine process of the maxillary bone here as well, forming the floor of the nasal cavity. And this bony floor is known as the hard palate. Okay, so that's the structural architecture of my nasal cavity. We mentioned the conchae. There are, of course, a total of 6 conchae, 3 on each side. So I've got a superior nasal concha right here, a middle nasal concha, and an inferior nasal concha. The superior and middle nasal conchae, they are part of my ethmoid bone. The inferior one is a bone unto itself. All of my conchae collectively work to give me more surface area and to create bumpiness that will allow airflow to be more turbulent. More surface area means more mucus, more turbulence means more time. That combo is good for better filtering, warming, and humidifying of the air. By the way, notice the nasal septum that I can see here as well. I got my perpendicular plate of my ethmoid bone up here. I got my vomer down there. Okay, on that note, let's keep going. We can see some nice conchae in this nose as well. All right, if we slice a nasal concha, so I got a slice of a nasal concha right here. So here's the nasal cavity space. This is a nasal concha right here. And I want you to just notice all the vasculature in here. A lot of blood vessels within the nasal conchae. Very small distance between the air space and the blood space here. That's going to make it easy to warm up the air that we're breathing in. And of course, the mucus is going to help with the humidifying and the filtering. Okay, we leave the nasal cavity through passageways called the posterior nasal aperture. There is a posterior nasal aperture right there. There is another one right there. Okay? I just made a terrible arrow there. Let's get rid of that. There's a better one right there. So these are my posterior nasal apertures. They're basically where this yellow line is, separating my nasal cavity from my nasopharynx. And of course, this is anatomy class, so we have multiple names for the same thing. The posterior nasal apertures are also called the choanae. Whichever name you like better is the name you can use. All right, before we fully leave the nasal cavity, we would be remiss if we didn't include a little conversation about the paranasal sinuses. There are 5 paranasal sinuses. Remember, a sinus is a cavity within a skull bone, and I'm going to have such a cavity in my frontal bone, my ethmoid bone, my sphenoid bone, as well as my left and right maxillary bones. I can totally see my frontal sinus right there. I can see my ethmoid sinus right there. I can see my sphenoid sinus right there. I cannot see my maxillary sinus in this sagittal slice, but I can see 3 of them, 3 of my 5 sinuses right here. Now, the big function of sinuses is the production of mucus. That is the number 1 major function. Other functions include lightening the skull and creating resonance for voice production. Now, the mucus made in these sinuses has got to drain into the nasal cavity. So there will be passageways that allow mucus to go from the sinus into the nasal cavity. It's kind of like the nasal cavity isn't big enough to make all of its mucus on its own, so it's got these mucus factories, these like off-site mucus factories. All right, let's take a look at our maxillary sinuses. So we need a different slice. So we did a frontal section through the skull here. And I can see the nasal cavity right in here. I can see my conchae, my inferior and my middle nasal conchae here. I can see my bony nasal septum with the vomer and the perpendicular plate. And now I can also see my biggest sinuses, My biggest sinuses are my maxillary sinuses. Here is one maxillary sinus on his right, and then the other one on his left. And I can even see the passageway that mucus would take as it comes up and goes into the nasal cavity. And of course, we not only drain mucus into the nasal cavity, we also drain tears. So we're always making tears. The lacrimal gland is always making tears. Tears flow across your eye, empty into these little ducts and into the nasolacrimal sac and the nasolacrimal duct, and then onward down into the nasal cavity. Okay, fantastic. Let's jump out of the nose and the nasal cavity and enter the throat, better known as the pharynx. There are 3 regions to the pharynx: naso, oro, and laryngo. Look at the uvula here. If you're basically next to the uvula, you're in the nasopharynx. Check out the epiglottis here. If you're next to the epiglottis, you're in the laryngopharynx. Then if you're in between the uvula and the epiglottis, you're in the oropharynx. So that's how we know where we are. So let's talk about that nasopharynx first. The nasopharynx is the superior part of the pharynx, going from the posterior nasal aperture all the way down to the tip of the uvula. Soft palate is here. Uvula is the hangy down part. The lining of this cavity would be pseudostratified columnar epithelium, because this is a respiratory only passageway. Prominent structures include the pharyngeal tonsil, which is on the back here, as well as the auditory tube, which is a passageway right there. The auditory tube, we see it nicely in this picture, connecting the nasopharynx with the middle ear cavity. The idea is we got to make sure the air in the middle ear cavity, which is the space behind your eardrum where you have those 3 little bones, we got to make sure the air in there is at the same pressure as the air around us, and this isn't a route for air to flow in or out to balance that pressure. We certainly become aware of that pressure difference when we are on an airplane. Okay, let's jump down to the oropharynx. The oropharynx is that middle part of the throat, stretching from the uvula to the epiglottis. The lining here is going to be stratified squamous epithelium, because this is not an air-only passage, but food and drink goes through here as well. So we need the greater protection provided by stratified squamous epithelium. And we will have the palatine tonsils basically in that lateral wall right there. Okay. Let's zoom down to the laryngopharynx, the inferior part of the throat. So we had naso, oro, laryngo. Basically, if you're adjacent to the epiglottis before we have the division, so here's the larynx in the front, and then the esophagus is right here. So if you're before that division, after the tip of the epiglottis, you're in the laryngopharynx. Again, this is a common passageway: air, food, drink. So we need the protective epithelium, which of course is a stratified squamous. And now we're done with the pharynx. Let's move on to the larynx, or the voice box. It is a tube with a lid. There are 9 cartilages in it. It is primarily lined by a pseudostratified columnar epithelium, except, portions of the top. One of the other big jobs besides voice production is going to make sure food and air go down the correct way. We got to make sure that air goes down the anterior tube and food goes down the posterior tube. And that is what the lid of the larynx is for. This lid, the epiglottis, is going to shut when you are swallowing food and drink. And that makes sure food and drink don't go down this passage. All right, here I can see some of the laryngeal structures. So I do have my trachea below my larynx, got my hyoid bone attached to it and nearby. My biggest laryngeal cartilage is my thyroid cartilage. Right beneath it, I've got my cricoid cartilage. The middle of the thyroid cartilage is where the Adam's apple is. That is a very prominent part of the larynx, so it is appropriately called the laryngeal prominence. I also see the epiglottis standing up. So I see three of my major cartilages here. By the way, the epiglottis, of all of the cartilages that we're going to see, the epiglottis is the only one made of elastic cartilage because it is the one that we move the most. It is the one that changes position the most. That needs to be, to be bent and then to snap back to where it originally was. My other cartilages, thyroid, cricoid included, they are made of good old hyaline cartilage. Okay, let's keep on going. If we turn this guy around, I still see my thyroid cartilage. Notice it does not span the back. The word thyroid means shield-shaped because a shield would not encircle someone. Cricoid does go all the way across. The cricoid refers to a particular type of ring. Got the epiglottis sitting up. And I do see my other three cartilages as well, my arytenoid, my corniculate, and my cuneiform cartilages. Now, everybody but the epiglottis is made of hyaline cartilage. The epiglottis is made of elastic cartilage because it is the one that is folded, that is used as the lid to the larynx to cover it up when we swallow. Slicing the larynx in half mid-sagittally, I see my hyoid bone right there. I see my thyroid cartilage. I see a little bit of the cricoid down here and a lot of the cricoid in the back. I see the epiglottis sitting up. Of course, the epiglottis would fold down this way to cover this opening. And when I slice it like this, I do see a pair of folds. There is the upper vestibular fold and then the lower vocal fold. The vestibular fold is a protective layer that's covering the vocal fold, which is where you have your true vocal cords, which I am using to do all this speaking. All right, I just paused this video to have a drink of water, and I can assure you my epiglottis was working because I did not choke on it at all. In case you were wondering, in case you were worried about me. If I take a look down superiorly, I can see the glottis, which is the opening in the space between the vocal folds. I can see my vocal folds right here, the whitish guys, the vestibular folds above them. I can see it in here as well. And what I want you to notice too is we can vary the amount of space in our glottis. We can make it big, we can make it small. That's going to help me adjust the volume of my voice. If I want to speak more loudly, I'm going to use a wider open glottis. If I want to whisper, I'm going to use a narrow glottis. We can also close the glottis and have a sphincter function to increase our thoracic pressure if we are trying to lift something heavy or if we are straining because we're having difficulty pooping. We can close that glottis, and there's a sphincter function here which lets me increase and build up and maintain high thoracic pressure. All right, with that, we're done the larynx. Flying through some respiratory organs today. Let's talk about the trachea. And the trachea is my air tube. It runs from my larynx down to my main bronchi. Trachea has basic conducting zone functions. Do notice that the trachea has cartilage in it, hyaline cartilage, which keeps the trachea open. And the cartilage is C-shaped. It is not present in the back. Instead, we have a muscle called the trachealis muscle. And the reason for the absence of cartilage in the back here is to make room for this. This tube right here, do you know what it is? Of course, it is the esophagus. And if we take a big bolus of food and swallow it, the esophagus has got to be able to expand, and it needs to expand into the trachea. And if there were cartilage here, that expansion would not occur. So notice we have a ciliated epithelium. It's going to be pseudostratified columnar pseudostratified ciliated columnar with goblet cells. We've got seromucous glands underneath the mucosa in our submucosa here, where we're going to make even more mucus. And then we got our cartilage. And then on the outside, we've got an adventitia, an anchoring layer of connective tissue. So some good stuff in the trachea right there. If we zoom in on the histology of the trachea, we see that pseudostratified ciliated columnar epithelium with occasional goblet cells. Here's the submucosa. We've got glands in it, as well as blood vessels and connective tissue fibers. Then down here is our hyaline cartilage, which is giving the trachea support and preventing it from collapsing. And with that, we are done with the trachea. And I'm going to call it quits right there for lecture video number 1 on the respiratory system. So we're about a third of the way through. We'll pick it up next time. I'll see you then. Bye-bye.